This animal hunts without light, without sound, without smell, and it has been doing it for 400 million years. Off the California coast, at a depth of 1,500 meters, the water temperature hovers near 2 degrees Celsius. No sunlight reaches here, not a fragment. The pressure at that depth is 150 atmospheres, enough to crush unprotected equipment. And in that blackness, a Chimaera moves. It is not a shark exactly, not a ray. Hydrologus trolli, the pointy-nosed blue Chimaera, belongs to a lineage that diverged from sharks and rays so long ago that the split predates the first trees on land. Dominique Didier at Millersville University established this through molecular phylogenetic work published in 2012. 400 million years of separation. The animal alive today carries that entire history in its cartilage. Its eyes are large, flat, silver in ROV light, but at 1,500 meters, eyes are nearly useless. The Chimaera knows this. Evolution knew it first. Across the snout, arranged in lines, are clusters of pores, dozens of them. They do not see, they do not hear. They detect something else entirely, something the Chimaera was reading long before any vertebrate on Earth developed the machinery to see. In October 2009, an MBARI ROV filmed a living hydrologus trolli in Monterey Canyon for the first time. Researcher Bruce Robertson led that expedition. The footage showed an animal completely unbothered by the blackness around it, moving with purpose. Not searching, knowing. That footage raised a question nobody had answered cleanly. How does a vertebrate locate prey it cannot see, hear, or smell, buried under sediment, in water with no light, at pressure that defeats almost every other biological sensor? The answer involves a biological instrument older than the eye itself, documented, named, measured to a precision that should not be possible. MBARI logged a specific dive, a specific depth, a specific date, and what the ROV recorded that day, an animal doing something no vertebrate eye could replicate, is the thing this record was built to file. The date is the 18th of November, 2016. The depth is 1,500 meters. The easy answer is this. Chimaeras are deep-sea scavengers. They drift slowly across the bottom, nose down, following chemical plumes from decaying organic matter. Most bottom-dwelling fish work exactly this way. Olfactory detection, lateral, line vibration sensing, passive drift until something edible registers. Case closed. Except it isn't. The lateral line is real, and it works. Tiny mechanoreceptor cells running along a fish's flank detect pressure changes in the water, movement, vibration, the bow wave of an approaching animal. It is a genuine, well-documented sense. For most bottom dwellers it is essential, but it reads motion, specifically motion. Olfaction is equally real. A chemical gradient dispersing through water carries enormous information. Sharks are famous for it. Many deep-sea fish rely on it entirely. The assumption, reasonable on its face, is that Chimaeras do the same. Here is where it breaks down. In 2013, David Ebert at Moss Landing Marine Laboratories co-authored a comprehensive taxonomic review of Chimaera species. His team documented the olfactory capsules directly. Compared to the skull size, they are small, reduced. Not the architecture of an animal built around smell. Then, there is the footage problem. MBARI's ROV cameras have recorded Chimaeras hovering above undisturbed sediment, flat, featureless, no organic matter visible on the surface, no current moving through the water column. Zero chemical gradient dispersing upward, zero water movement to trigger mechanoreception. And the Chimaera still found what was underneath. The lateral line cannot explain that. No movement occurred. Olfaction cannot explain it either. No gradient existed. Two of the three tools in the standard deep-sea toolkit simply do not apply to what the cameras recorded. So the comfortable answer, scavenger, smell, vibration, drift, accounts for some behavior, and none of the specific behavior that needs explaining, which forces a harder question. If the Chimaera is not reading chemistry and not reading motion, What signal is it reading from a buried animal that has given nothing away? 5 nanovolts per centimeter. A standard AA battery produces 1.5 volts. Divide that by 300 million. You are now approaching the electrical field a flatfish generates simply by beating its heart, buried under 2 centimeters of cold sediment, invisible to every sense evolution ever built, except one. The pores run in lines across the Chimaera's snout. Adrian Gilte first described them in 1922, cataloguing specimens at the Leiden Museum. Each pore opens into a canal filled with a clear, conductive gel. At the base of the canal, sensory cells wait. When an electrical field passes through the gel, even one measured in billionths of a volt, those cells fire. The signal travels to the brain. The brain resolves it as location. Dominique Didier, in her 1995 phylogenetic study published in American Museum Novotates, described the structure. Alveoli connected to surface pores by long canals filled with a gelatinous substance, morphologically identical to the ampullae of Lorenzini in sharks and rays. The same architecture, the same gel, the same threshold. R. W. Murray at the University of Birmingham published the proof in 1960 inches Nature. His physiological experiments demonstrated that elasmobranch ampullae responded directly to electrical stimulation. Not chemical, not mechanical, electrical. Adrian Kalmine refined the behavioral picture in 1971 and 1982, documenting that cartilaginous fish could locate prey using bioelectric fields alone, with no other sensory cue available. David Ebert's team made it precise. In Sharks, Rays, and Chimaeras of California, published in December 2013 by the University of California Press, they stated it plainly. These fishes can detect electric fields as weak as 5 nanovolts per centimeter, allowing them to sense the bioelectric fields produced by hidden prey buried in sediment. The footage came next. The 18th of November 2016. An Embari ROV descended off the California coast to 1,500 meters. The pressure outside exceeded 100 atmospheres. No sunlight, no chemical plume. The Chimaera in the frame held its body motionless, not drifting, not searching, snout angled at the sediment. Then it struck. A single movement. The silt exploded outward in a small cloud. No instrument aboard the submersible had detected anything beneath that sediment. The Chimaera had. Chapter 1 left a question open. A researcher's hand, drawing a chalk line on a university lecture board, marks 2 points. 450 million years ago, electroreception. 350 million years ago, vertebrate vision. The gap between them is 100 million years. Chimaeras were hunting in the dark before any animal on Earth could see. Not as a fallback, not as a supplement, as the primary sense. The first complex predatory map of the world was not built from light. It was built from electricity. Every predator that followed, every shark, every ray, every lineage that inherited the ampullae entered a world already charted. The coordinates were electrical, the map was invisible. And the Chimaera was already reading it, alone, in the dark, before sight existed to compete with it. The index concludes this: the Chimaera's electroreceptive system is not a curiosity of the deep. It is the original predatory sense, older than vision, still functioning at 5 nanovolts per centimeter. The November 2016 Embari footage is simply the most recent confirmation of something that has been true for 500 million years.